Like a lot of people in the earth sciences, we're interested in hunting fossils. And look at a rock like that. It's an ammonoid fossil, about 150 million years old, relative of today's nautilus, squid, those sorts of organisms. Is very obviously a fossil. You can see the similarities between this rock and, or the object in this rock, the ammonoid, and contemporary organisms that we can find in the ocean. So paleontology is very clearly about preserved remains of organisms from the past. But the remains that we're particularly interested in are the molecular remains. So things that are much finer scale than a large fossil like this, and things that are much more intangible, I think, in terms of whether they're of biological origin or not. So the sorts of fossils that we're interested in are organic molecules. Typical organic fossil is something like this. It's a sample of petroleum from Saudi Arabia. It's what we're using to power our economy, to power our cars. It's composed of the remains of organisms that grew in the ocean, again, around about 150 million years ago. And it's very clear when you analyse the material in this particular sample that the molecules bear strong resemblances to molecules that we can find today in organisms. We can find the remains of photosynthetic pigments, for example. We can find the remains of membrane lipids. And our job as a geobiology and astrobiology laboratory is to focus on ancient sedimentary rocks and see if we can find molecules that tell us something about the organisms that lived in the past. And as we go further back in time, it becomes more and more difficult because the rocks have seen high temperatures, high pressures, they've been folded, they've been weathered, and they've been colonized by contemporary organisms. So it's a difficult process to disentangle what's ancient and what's modern. So one type of rock that we're particularly interested in, rocks that are grey to black, and those coloured rocks are the ones that we find tend to have significant amount of organic carbon in them. And it's the organic carbon that we're interested in analysing, dissecting, and then trying to reconstruct the chemistry of that organic carbon to what sort of organisms might have produced it in the beginning. This particular rock is a stromatolite. It's about 800 million years old, from Central Australia. And you can see very, very fine laminations. You can see that it's composed of objects that are sort of dimensions of a finger. And between those objects, you can see some evidence of ancient mud. This is a particularly good rock to study because from the package of sediments that it occurs in, we know that rock was formed in shallow water. We have a very good handle on its age, as I said, about 800 million years. And the fact that it's not twisted, bent, orange, tells us that it's not been subject to extreme weathering. Unfortunately, the rocks that we would most like to study are really, really old rocks. And you can see in this case that those rocks are distorted, they're oxidized, they're broken, they're weathered, they've probably been colonized several times by more recent organisms. It's very, very difficult and problematic to get chemical fossils from a rock like this. But there are small amounts of organic carbon, and we can get their isotopic patterns, and from that we hope to be able to at least say something about the likelihood that this was formed by biological processes or otherwise. In some cases, we have an intermediate situation. So this rock here is also very old. It's 3.5 billion years. And you can see, in this case, the rock is not weathered. The laminations are very well preserved. So this is a target that we're currently investigating, hoping to isolate and characterise some of the oldest organic matter that's been discovered on this planet. This particular rock here now is of very great interest to many, many geologists, geobiologists and astrobiologists. It's a piece of banded iron formation, and the current dominant theory about how rocks like this formed is that they were produced as a consequence of iron that was liberated from subsea volcanoes in its reduced form, iron 2, coming in contact with oxygen produced by photosynthetic organisms living at the surface of the ocean, and essentially that iron going rusty, becoming insoluble and sinking to the bottom of the ocean, initially as a sort of jelly-like substance, and subsequently becoming lithified or forming a solid rock. And this particular rock has very, very fine banding. If you see this in the field, those bands go for tens to hundreds of kilometres uninterrupted, telling us that this was deposited at the bottom of a deep ocean, undisturbed by the action of waves, undisturbed of course by the action of biology, because complex life didn't exist at this time. But this is a key piece of evidence, or rocks like this are a key piece of evidence for changes in the oxidation state of the surface of the Earth through time. We know that the Earth was formed without any oxygen or any minute traces of oxygen in the atmosphere, but today we have 21% oxygen. And it's a key objective of the NASA Astrobiology Institute teams to understand how this process happened. What were the original organisms that learned how to produce oxygen from water? When did they learn how to do that? Were they efficient? What happened to keep the oxygen in the ocean atmosphere system low for at least 2 billion years before the advent of complex life, which we use as a sign that the atmosphere and ocean had roughly similar contents of oxygen to the ocean atmosphere today, about 21%. But the course of the planet's oxygenation from going from essentially zero oxygen to 21% oxygen, it's one of the greatest enigmas and the greatest problems in geobiology today. There are a number of very, very important consequences for the trajectory of life that are related to the oxygen in the atmosphere-ocean system. And of course one of them is oxygen. When you use oxygen to burn sugars, you achieve a lot more energy from that process than you would if you fermented that sugar to form alcohols. That's a low energy producing process. Burning with oxygen is a high energy producing process. And complex life that moves, life that thinks, couldn't exist without the aerobic respiration, or the energy that we get from aerobic respiration. Another consequence of the change in oxidation state of the Earth's surface is the availability of trace elements that are so important for biology. The availability of iron, molybdenum, nickel, those elements that are so important to our enzymes, certain key enzymes, their availability changed through geological time with the increasing oxidation state of the Earth's surface. So there are many, many ways in which oxygen has impinged on the trajectory of life, and it's a very exciting research activity to work with lots of other people who know about biology, people who know about genomics, people who know about geochemistry and geology, the age of rocks, putting all those small snippets of information together to reconstruct as best we can the history of life on Earth. So the rocks that I've been showing you up to now are rocks that come from different parts of the Earth. Quite a number of them come from Australia, and there's a natural reason for that. That's because I come from Australia. I'm very interested in Australia as a repository of life history. It's very difficult to see because it's in this glass vial, and I don't wish to open the vial. It wouldn't be good for the rock. But the rock in here is very, very important, of a class of rocks that tell us something about organic chemistry before the origins of life on Earth. So this is a sample of meteorite that fell near Melbourne in the 60s. It's a Murchison meteorite, and it's composed of material that's considered to be primordial, that is, representative of the material from which the Earth formed. So it contains the elements iron, silicon, oxygen, carbon, etc. But it also contains carbon compounds that have been formed in interstellar space, and it's thought that these carbon compounds were delivered to the Earth in much greater volumes than they are today, but when the Earth was being pounded by impactors, there was a source of organic carbon compounds provided to the planet from the interstellar medium. And for many years, people have been looking at rocks like this, looking for evidence of molecules related to those which we use in our cellular metabolism today. And this rock contains amino acids, it contains nucleobases, it contains organic acids, dicarboxylic acids, hydroxy acids. It contains a lot of compounds similar to those that are used by biology, but not identical. And one of the great questions for us as scientists today, and I guess the general public who are thinking about where we came from, is what were the stages or what were the transitions required to turn chemistry, as represented in this rock, into the wonderful biology that we have around us today? It's a great question, and it occupies a lot of our time and a lot of our thinking. And we're very grateful actually to the Simons Foundation who have instigated a funding activity called the Simons Foundation for the Simons Collaboration for the Origins of Life. And this collaboration involves people from all over the US and from Europe, and allows us to research the primitive chemistry of rocks like this, the early history of life on Earth as represented by rocks like this, in the hope that we can learn more about our origins and the possibility of life beyond the Earth.
So in this laboratory, we're very interested in the chemistry of rocks. We're interested in the chemistry of rocks that are being formed in the bottom of the ocean at the present time, but we're also very interested in rocks that were formed at the bottom of the ocean in ancient times. In particular, this one here, for example, is 3.5 billion years old. It's a black chert. People have found microscopic objects in here that they think are fossils, but they don't look like any microbes that you would find on the Earth today. So there's a lot of controversy about it. What we're trying to do is extract chemicals from this rock that might support or disprove the idea that the objects that have been found are actually fossils. So we're doing chemistry, focusing on chemistry. On the right side of the lab here, you see a row of fume hoods. And because we're handling hazardous chemicals, all the work with hazardous materials has to be done in these fume hoods. And the fume hoods are allocated to different purposes. Some things we do very, very clean work, or we try to do clean work, and some of the hoods have acids, very strong acids, very dangerous. So we partition the different tasks to the different hoods. But this side of the laboratory is mostly about preparation of samples, isolation of materials, minerals, organic matter, that sort of thing from rocks like this. The other side of the laboratory, you may be able to hear the noise, are all analytical instruments. So each one of these instruments that you can see on this side is a mass spectrometer. So altogether, we have 6 mass spectrometers in this laboratory. The one to my left here is called an isotope ratio mass spectrometer, and we use it to measure the amount of carbon-13 and nitrogen-15 in natural materials, particularly in substances that we can extract from rocks like this. And the relative amount of carbon-13 can tell you something about the processes that led to the formation of that organic matter. In some cases, we can use it to distinguish between processes controlled by biology versus processes that are purely chemical. So we're in particular looking for the carbon isotopic ratios of different materials measured on this machine here. This is probably our biggest mass spectrometry instrument. It's called a magnetic sector mass spectrometer, and it's configured to do what we call analog experiments. And we're funded by NASA at the moment, by the NASA Astrobiology Institute and Mars Science Laboratory Participating Scientists Program to do experiments in the lab here that mimic, as best we can, experiments that are currently being conducted on the surface of Mars by the Curiosity rover. The Curiosity rover can drill into the sediments on Mars, get small amounts of sediment from the surface rocks, and heat those sediments in a stream of helium gas, inert gas, drives off any volatile molecules and analyzes them using a mass spectrometer. So this device here is, we call it a pyro probe, but it mimics the sampling system that SAM has on Mars. So we have little tubes with a tiny amount of rock go into these holes here. The tubes drop into an oven. That oven is heated at different rates, sometimes very fast, sometimes very slow. And as the gases are liberated, they come through this pipework and ultimately find their way into this part of the instrument, which is a gas chromatograph. And the gas chromatograph is the basis of separating molecules one from the other so that we can subsequently identify them unambiguously using a mass spectrometer. This is the working core of the instrument. It's a gas chromatographic column. You see it's a very, very thin tube made of fused silica. It's 30 meters long, and it's got a coating of liquid on the inside that performs a separation of the molecules that we're analysing. And on Curiosity, on the SAM instrument, there are 6 of these columns with different qualities that allow it to do different kinds of separations. But this column in here, at the present time, is the one that's identical to the ones that are being used so far by the SAM instrument. This is a spare column. We have lots of columns in our laboratory. We can change them according to the sorts of experiments that we wish to do. But this particular type of column is one that was used for the first detection of organic molecules that we think, or we're pretty confident, that the carbon in those molecules is of Martian origin. So this is a very large instrument. SAM is itself a bit bigger than a suitcase. So the instrument on Mars is a miniaturized version of this. But the advantage of having a machine as complicated as this, we can vary the conditions of the experiment according to what we find on Mars, improve the quality of the experiment, improve the fidelity of the experiment, provide information for the next sort of planning stage, how the next experiment is going to be done on Mars. One of the important things about making sense of the results that we get from Curiosity is whether or not the experiments are reproducible. And a key element of the analog experiments is that people in different laboratories can do the experiment in the same way and get the same result. So there are people at Johnson Space Center in Houston, Texas. There are people at the Goddard Space Flight Center in Maryland currently doing similar experiments on similar instruments like this. In particular, at Goddard Space Flight Center, they have a version of SAM that's almost identical to the instrument that's flying on Mars, and they operate it under Mars-like conditions in a chamber at low atmospheric pressure at Mars temperatures as close as they can, mimicking the conditions that exist on Mars. That instrument, being in a vacuum chamber or a very low atmospheric pressure chamber, that instrument's complicated to use. It requires the same kind of loading software, loading samples as happens on Mars. So it's limited in the number of experiments it can do. It can do perhaps 3 or 4 experiments a day. We can do hundreds of experiments in a day, as can people at Johnson Space Center. So what we lack in fidelity of operating under Mars conditions, we can make up for in being able to do many, many more experiments under quite different conditions, and therefore build up a set of information or a set of data that helps us interpret the results that have gone before, but also plan experiments for the future on Mars so that they will be conducted to achieve the most important information or get the most important information from any particular sample. And this also involves working closely with the other team members on Curiosity, the geologists, the geochemists, the physicists. It's very much a teamwork experience. None of us know enough to do an experiment in its own right, but working together from different specialties or coming from different perspectives, we can maximise the value of the experiments done by Curiosity. As you can see around us, there's a lot of very, very expensive scientific equipment in this laboratory, and a lot of people operating that equipment. In order to maintain a laboratory like this, we're very dependent on our funding sources. Probably the major funder of equipment and people in this laboratory is NASA, through the NASA Astrobiology Institute, where we have one of the lead teams with the focus of that research is partly connected to the search for habitability on Mars, but also the search for evidence of increasing complexity of life on this Earth. We're particularly working on the advent of animal life, complex multicellular animals, and how they came to be, what conditions of the Earth existed that allowed complex life to form and to flourish, and how that complex life in itself changed the surface of the planet. Life and the planetary evolution are so delicately intertwined, it's impossible in some cases to separate the effects that are biological versus the effects that are chemical. So a major focus of our research is understanding how complex life came to be and how complex life has changed the planet forever. We're also interested to a degree in the future of life on Earth, what we're doing to the planet today that may make it difficult for complex life to continue into the future. So the NASA Astrobiology Institute is a key source of funding. We also rely on some private foundations. Currently we're being supported by the Simons Foundation, collaboration on the origins of life. So in that project we're looking at the earliest stages of life on Earth and the earliest evidence for life on Earth. We're also being supported by another private foundation called the Agron Institute. And the Agron Institute have been particularly important for funding drilling projects on the Earth that allows us to get very fresh samples from critical intervals of time in South Africa and most recently from Australia. So combinations of funding sources are what allows us to keep doing our work, and continuity of those funding sources is very important for people's careers. When a student enters our lab, for example, they're going to be here for 5 or 6 or 7 years. We need to be making sure that those funding sources are stable for a sufficient amount of time for people to do their work and bring it to a fruition. That means conceiving the project, conducting the research, and then writing up the study and presenting it to our colleagues for scientific criticism. Scientific criticism means not saying you're wrong or right. What scientific criticism is about is testing, continually testing your results for their reproducibility, finessing experiments so that you are able to be absolutely confident the result is correct. And many experiments are required in order to be confident in the result and have your colleagues be also convinced that the result is robust.
So once upon a time, if you wanted a career in science, you went to graduate school and got a Ph.D. and got a faculty position, and that was that. And that's not the situation anymore. There are very, very few tenure track faculty positions relative to the number of students who begin a career in the sciences. And so the postdoc is an important part of that training process. It's a part of that pipeline. Nowadays it's pretty much standard for PhD graduates to do a postdoc to receive further training either in their field of specialization that they were in for their PhD program, or in my case, it's been a great opportunity to learn a whole new skill set and to sort of expand my skills and experiences. So my background is actually in paleontology. I trained as a geologist and did my PhD research on what we can learn about the process of evolution from microfossils. And since coming here, I've been able to undertake a big transition to working in a really different field of organic geochemistry. And so I've been involved in the Curiosity mission analog work that we do in the lab here. I've been able to learn a whole new skill set of operating the instruments that we use to conduct those analog experiments here, making synthetic samples of Martian sediment and analyzing them to help us better understand and interpret the data coming from the Mars rover and the experiments that are being carried out on Mars. I'm very fortunate to have a second role in my job here at MIT alongside research as an educator and a science communicator. And so through the funding that we receive here as the lead institution for a NASA astrobiology institute, I am responsible for coordinating the education and public outreach efforts of this institute, and so I get to work in collaboration with lots of different organizations in Cambridge and in the greater Boston area, and also across the country with partners in the NASA Astrobiology Institute to bring the science and the research that we're doing to the general public through the education system and also through informal science education efforts. So, for example, we work together with the Science Club for Girls here in Cambridge. We work with the MIT Museum to develop a presentation series, their evening discussion series, working on exhibits at the MIT Museum. We also have a long-standing collaboration with partners at Arizona State University on a series of virtual field trips. These are online learning experiences that allow students in high school classrooms, middle school classrooms, but also the general public to experience field locations of astrobiological interest that you otherwise wouldn't be able to get to, that are inaccessible, that are far-flung in all different continents, and so I feel that it's equipped me with a skill set that's very useful in my research. It's very useful for education, but it's also a broad general skill set that I think is going to be useful in the rest of my career, wherever that takes me.